Alright, so let's get started. So we'll be discussing about absorption by roots. So we'll see the different processes that are involved, like imbibition. So there is osmosis, there is a diffusion. So in osmosis again there is exosmosis and endosmosis. Okay, then we'll see what is active transport, the turgidity, flaccidity, the plasmolysis. So basically I'll be concentrating, I'll be highlighting these terms in today's session. All right. So before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology and the Unacademy channel, that is ICSE and ISE both. So this link you can find in the description box. So you can click on the link and join the channel for updates regarding the classes. Also, we conduct special classes on Unacademy platform, which are actually absolutely free, because here classes which are done, so these are specially named as special classes. So that is because you have an option to learn from any educator, so that is for free, and a certain percentage of topics like a syllabus will be covered in these classes. All right, so in that case you can engage with educators via poll, clear your doubt in the live special class. So we'll be conducting a quiz there, so you can participate. We'll be sharing the leaderboard, so you can know your rank and compete with your friends. So clear your doubts in the live special class. So there is a question tab separately where you can type in your question, attach an image and send it to the educator during a live session. They'll be getting a notification and will be clearing your doubt. So now for the top special classes, you can visit the link in the description. All right, so Unacademy subscription features, so these are plus class features. Okay, so learn live from the comfort of your home. Unlimited access to all the courses will be there, different educators will come up with their own courses, so you will have access to all the courses once you take the subscription. Then top educators of India are going to take the classes in one platform, that is Unacademy platform. There is regular doubt clearing session that will be done. Okay, so that is up, after about every 3 sessions, the fourth session will be our doubt clearing session. So whatever doubts you have related to the topics that are previously covered, so you can clear your doubt in this session. Okay, then complete syllabus will be covered. Mentorship and guidance will be given. Study material is given, so that is in the form of PDF. Then practice test is given. Live test series will be there. So, and also there are batch courses. So basically, so you will have access to English batch, Hindi batch. So once you take the subscription, so you will have access to all the batches. So depending on your preference to learn, you can go for any of these batches. Okay, so then with that, there is a daily practice section, weekly mock test series will be there, live quizzes will be there, then there is daily MCQ and subjective test series are also there. So this is a unsat, Unacademy national scholarship admission test it is. So this is a offline test for IIT, JEE, that is JEE, then NEET UG, and also class 9 and 10. So this is conducted across 40 cities, and the test is on June 4th and 5th. Okay, so you can get up to 90% scholarship worth 150 crore for Unacademy centers and online subscriptions. So this is basically offline classes also were being conducted. Okay, then top 40 rankers win an iPhone. So basically test takers get an Unacademy t-shirt as well. So you can enroll now, and for registrations you can the last date is basically June 2. Okay, so that is June 2nd, and 199 is the enrollment fees that you can get, and on top you need to use the code VANIYT. So with this code you will be getting 10% discount on the subscription. So that means you can enroll, and the link for this unsat you will find in the description again. So go to the description box, you will find the link in there, so you can just join in that. All right, so Unacademy plus subscription pricing. So now for 12 months of subscription, so since this is the very beginning of your class 10, okay, so you can go with the 12 months of subscription. Like for 12 months you can see here it is 17,250, where you will be like with the code, like you need to use this code here in the gift box. So that is Vani YT to get a 10% discount on the subscription. Okay, so you can also just try with 3 months or maybe 6 months, so depending on your comfort, so you can even go for the higher month. So this is completely your choice, and you can go for any of this one. Okay, hi Jinder, good afternoon, good evening. Okay, so let's see this. So we'll be discussing about the absorption by routes. So now let's say you are starting a new chapter to study. All right, so in that case what you can do is, so first you need to have a look at the syllabus, and this is important. Like every time you open up a new chapter you are planning to study, so you need to make sure that you are covering the points from the syllabus, you are covering the points from the scope of syllabus. Okay, so there is nothing that is coming out of this box. Okay, so even if it is coming, so basically they would give a grace marks, or otherwise basically these things make sure that you're perfect with all these things mentioned here in the syllabus and scope of syllabus. So this would do, like this is more than sufficient. So once you are thorough with this one, so you can go with the next chapter, like when you are studying a chapter wise. So in that case, so make sure that you follow syllabus as well as the scope of syllabus. Yes, so there is a reduced syllabus. So I think evolution chapter, only evolution chapter is removed. Rest of the things are same. Okay, only the evolution is removed. So the rest of the chapters are there. Now again, depending on any pandemic, if there is anything as such, maybe there might be changes, but as of now it is only evolution has been removed. Rest of the things are all there. Okay. Okay, so now let's get started. So this is with respect to absorption by the root. So what exactly happens? So absorption by the root is, so you can see that the water is being absorbed. Okay, so there is the absorption of water, you can see. So not just the water, so along with that the minerals are taken. So any idea why exactly water or a mineral is required for a plant? Because we'll be discussing about, so mainly in today's session I'll discuss the terms like imbibition. So I'm going to discuss about all this session, so I'm not discussing the characteristics of root hair and all. So today's session I'll be discussing the terms like imbibition. So there is osmosis, then there is diffusion term is given, the active transport is given. Okay, so we'll discuss all of that. The active transport, there is a term called as turgidity, then there is a flaccidity, then plasmolysis is given. So we'll discuss all those terms. Okay, so here the water and mineral nutrients play a major role for all these processes to occur. Now from all these processes only basically the transport of water, like water is being conducted, nutrient is being conducted. So that is because of these three, I mean these all these processes. All right, so let's get started then. So root fix the plant in a soil, giving it support, but the most important life supporting function of is of root is the water absorption and the mineral absorption. At a time water is being absorbed, let's say this. Now water is taken, so along with the water, so there are minerals also that are being absorbed. Okay, so now there is the plant cell. So this plant cell is taking that water. So there is a cell wall, then you can see a cell membrane that is there inside. Okay, so what we say, this particular cell wall, what is there? So this is actually freely permeable. So this is what? So this is freely permeable. That means what? it will allow the water inside, okay, and also it allows the minerals inside. So minerals are also allowed inside. So you can see minerals that are present here surrounding this area because it is freely permeable, okay. Then you can also see that there is a water that is being absorbed. But if we are talking about the semi-permeable membrane, so what is that semi-permeable membrane? So that is the cell membrane. So here you can see, like, water is completely present here, okay. So whole this thing you can find a water along with all the other minerals. Now this particular membrane, what you see, so this is the cell membrane. So this is not freely permeable, so this we can call a semi-permeable membrane. So let me just erase this, okay. Yeah, so this one is what? This is semi-permeable membrane. That means everything is not allowed. You can see this blue one. So these are water and these are minerals. So minerals will remain in here. But if a water is anyway freely allowed inside. So that is why we call it as a semi-permeable. That means it allows the solvent to move in freely, but the solute is not allowed to move in freely, okay. So solute is not allowed, but the solvent can move in freely. So you can see all of this water which is here, the blue one. So this can get in there, but the minerals which I have shown in a green one, so that is not moving easily, okay. So if a mineral has to be taken inside, so in that case active transport process has to occur, okay. So this is mainly there for the transportation of a solute or a mineral, or we can say ions, from the outside to the inside. So that we can say from low concentration to high concentration. So we'll discuss all these terms, okay. Hi Ananya. So there is a lot of confusion in cell cycle chapter. So which part? Actually I have already taken phases of cell cycle on YouTube, okay, and the structure of chromosome is also done. So good evening, please make a live session for cell cycle. So cell cycle it is already done, Harjinder, so you can just watch the recording, and yeah, one more session I'll conduct, so that is live session on cell cycle, okay. So that I'll do, okay. So let's see this. So there are different processes that we will come across. So let's see each one of these one by one. So that is absorption and conduction of water and mineral. Now if a water and mineral has to be taken inside, so in that case imbibition is a must, diffusion has to occur, osmosis has to occur, active transport has to occur, and so this is alternate, like root pressure. When you study, so you will study about the turgidity, flaccidity and other things, okay. So we'll discuss each of these terms one by one, all right. Yes, so we'll okay, so the name of that session is phases of cell cycle. I have given the name as phases of cell cycle, so you can watch the recording, but anyway I'll conduct one session on cell cycle, all right. So now entire mechanism of water absorption and a mineral transport. So this is basically depending on the imbibition. Any idea what exactly is imbibition? Any of you here? So what is imbibition? So there is a term called as imbibition, okay. So this is what actually, so imbibition process will occur. So that is for the absorption and conduction of water. So in that case, so there is a surface attraction that you can see, okay. So surface attraction you can see in this one. Imbibition is a phenomenon by which living or a dead cell, so these are the keywords, okay. So these keywords should be there, okay. So this is also a keyword, so that is the surface attraction. So living, dead, surface attraction. So while you are writing the definition, so these keywords need to be included, okay. So imbibition is a phenomenon, it is not just applicable to a living cell, so this is even applicable to a dead cell, okay. So in that case they are absorbing the water. So now let's say there is a door, one second. So now let's say there is a door, okay. So this is during rainy season, what happens to the door, okay. So now this is the door, if we say, okay. So in that case this door, you can see a swelling, okay. So this is basically it will absorb the water. So this is mainly during the rainy season. So it will take in as much, like as it is continuously coming in contact with the water. So water is absorbed by the dead cell, because this is a plant. Door is basically what? So this is a door is a plant. It is basically made from a plant cell itself, right? So that is plant itself. So in that case, you can see the plant cell, and that is the dead plant cell, which is taking in a water as the cell wall which is there. So that is having a capacity to hold more and more water. So in that case, the cell wall is freely permeable, so which will make the cellulose which is there. So you might be having an idea that cellulose absorbs water. Okay, so in that case, due to the surface attraction, so there is, let's say, not here anyway, the water won't go. So in that case, let's say water is over here, okay, and this door is closed. So in that case, as the water comes in contact with this door, so maybe we can show the water molecules here. Okay, so as the cell wall is freely permeable, so this will get in, and all the cells of the door, what are there, which are basically a dead cells. Okay, so they will absorb this water, and there is a swelling that you can see, like basically, so these cells are distended, they are expanded. Okay, so that process is called as imbibition. So that is because it is occurring because of a surface attraction. So as there is a surface attraction, so you can see a swelling in the door. So this is one example. So you can also see, here one more example. So that is, the what is it, resin, the what do you call in dry grapes. Okay, so the dry grapes, what are there, so they also swell up. So if you soak them in a soil, I mean not soil, sorry, if you soak them in a water, so basically they are absorbing a water, and you can see, so earlier this was the a resin that was there. So resin is basically, you can say, a dry grape itself. So after it starts to absorb the water, so you can see a swelling in that resin. Okay, so that is also occurring because of the surface attraction itself. So there again, living cells are not there, but it is a dead cell itself. Now all examples that I am giving, so these are of dead cell only. Okay, now if we have to mention with respect to living cell, so in that case, first, so let's say this is a root hair. Okay, so this is a root hair, which is basically a epidermal cell extension itself. So this is a epidermal cell wall. Okay, and one second, so you can see a cell membrane there. All right, so in that case, water which is there surrounding, so first it will get attached. So there is a surface attraction that you can see. So these are all water molecules, and along with that you can see associated minerals will also be there. Just a second, where is it? Yeah, so this one. Okay, so in that case, you can see associated minerals are also there. So you can see the minerals which are dissolved in the water. So in that case, you can see that the cell wall is freely permeable anyway due to surface attraction. So there is a imbibition, and then there is osmosis. So that means as the surface attraction is there for the cell wall, so now these water molecules will move in. Okay, and once that is done, now for this water to move inside this cell membrane, anyway all this water molecule along with the minerals, so this will freely, like aram se it can come in. So that is into this cell wall. So that can be because of the what is it, imbibition. Okay, so that is the surface attraction. Here for this water to move inside this cell membrane, so osmosis process we call. So that means here the surrounding condition should be hypotonic, and inside it should be hypertonic solution. Then only you can see a movement that is occurring. So water will start to move from hypo solution to a hypertonic solution. So what exactly is hypo or a hyper? Anybody? So ma'am, please tell when will be the live session for cell cycle. Um, okay, I'll assign one session. Maybe you can join in my community. So the community link you can find in my description box. So this video description box you can find a community link. Okay, so that I'll just update that. Okay, so update regarding the classes. So phases of cell cycle, I'll just decide it. So maybe in June month. So since only 2 days are left. Okay, so 30 and 31st will be done. So June 1st, maybe I can keep it on June 1st. Okay, or maybe June 2nd. So 1st or 2nd, I'll keep the phases of cell cycle. All right. Hypo is less and hyper is high. So what is that high and less? Hypo is less, yes, that is right. Hyper is less. So what is less? So that is low concentration or low concentrated solution, or in other terms we can mention that hypo, when we mention, you can find the low concentrated solution. So what is that low concentrated solution? So that means it is a dilute solution. So dilute solution means what? The water concentration is high but the solute concentration is less. So I have seen lot of students who get confused always, like between hypo and a hyper. So in that case, so we use the term low concentrated solution to high concentrated solution. So this can be used, or the Selena definition says that so water movement, so that is from their region of higher concentration to their region of lower concentration across a semi-permeable membrane. So this is the definition that is given for a osmosis. So in that case, low concentrated solution is also same, that is a dilute solution. What exactly is dilute solution? That means you can find more of a water and less amount of a solute. So you can write it this way. Okay, and hyper, when we mention, so this is actually a high concentrated solution. So we are mentioning a solution here. So solution means solvent is also there, solute is also there. So we are not just talking about a solvent here, so we are talking about a solvent as well as a solute. So that means high concentrated solution. So in that case, so you can say here high concentrated means what? So it is a concentrated solution. That means solute concentration is more but the water concentration is less, according to the Selena definition which is mentioned in there. Yes, so in that case what is mentioned is, so you can see that the water will move from its region of higher concentration to its region of a lower concentration, or the other definition which is mentioned, the water molecules will move from their region of a higher concentration to their region of a lower concentration across a semi-permeable membrane. So that is a cell membrane. Okay, so this we call as a osmosis. Another definition what we can write for osmosis is the movement of water molecule from low concentrated solution to a high concentrated solution across a semi-permeable membrane can also be mentioned here. Is that clear? Any doubt? As compared to salta, hypotonic. Yes, that's right. Okay, so in that case this is about the imbibition. So these are the different terms. So let's have a look at the diffusion now. Okay, so in case of diffusion, so what exactly is diffusion now? Okay, so we have seen a diffusion can, it's not that diffusion is restricted to only solvent. It is not that it is restricted to only solute. So diffusion we can see in gases, diffusion we can see in solids, diffusion we can see in the liquids. Okay, so here there is no barrier, so there is no semi-permeable membrane. So we are not talking about a semi-permeable membrane. So basically what is happening is movement of any molecule, be it a gas, be it a solid, solute, be it a solvent. So in all these cases the movement you can see from higher concentrated area, that is from high concentration to low concentration, basically the movement can be seen. So you can see one example which is mentioned. Okay, I'll just draw that. Okay, so let's say this is a beaker. Okay, and in this beaker basically you can see this beaker is completely full of water. Okay, so this beaker is having what? So this is having a water. So this beaker is full of water. Okay, So now you know in this beaker, so what exactly we have done is, so this is a water now which is having a particular, let's take potassium permanganate. So this experiment is given in your book. Okay, so this is for what? This is for a diffusion now. So I'm just trying to explain a diffusion process. Okay, and here at one corner, so what they have kept is a potassium permanganate is kept here. Okay, so this is what, so this one is a potassium permanganate. All right, so this one is potassium permanganate. All right, so this one permanganate. All right, I'll just write it as such. Now what will happen, so there is a diffusion, so gradually this potassium permanganate, so it will start to diffuse. Okay, and it will try to cover this whole area. So where exactly do you find it? So this is now a solid, which is here you can find a high concentration of potassium permanganate, and here it is low concentration of what? Low concentration of potassium permanganate. So in that case you can see that this potassium permanganate will start to diffuse so throughout. So basically you can see this will start to diffuse in all this area, and you can clearly see that this potassium permanganate will start to spread. Okay, so this will start to cover this whole area. So in that case let me just explain you this. So it will start to cover this. Now this whole water is actually covered, like this whole thing is now spread with the potassium carbonate, and now we have this whole water which was this. So this is completely pink in color. Okay, so this can happen. So this is only when a diffusion occurs. So in that case you can see the movement from high concentrated solution to a low concentrated solution. So diffusion is the movement, or it is a free movement we can say. All right, so free movement of molecules of a substance, so that can be solute, a solvent, a gas, liquid, from the region of their higher concentration to the region of their lower concentration when the two are in direct contact. So this we call as a diffusion. All right, any doubt in this? Okay, so here a sugar cube, or a potassium permanganate is being kept. So this example you can use. One more thing is, now let's have a look at the osmosis. So what exactly is osmosis? So I'll just explain you this. Okay, so we'll see. Okay, so now osmosis, when we mention, so this is with respect to osmosis, the definition that is given in a Selena. So this is taken from the Selena textbook. All right, so osmosis and osmotic pressure. So in that case, osmosis is the movement of water molecules from their region of higher concentration to their region of lower concentration through a semi-permeable membrane. Okay, so in that case you can see the water movement. So let's see, there is a hypotonic solution. So what exactly is hypotonic solution? And that we come across, like tonicity. So tonicity is there. So under tonicity you can find there is a concentration gradient. Okay, so tonicity is nothing but a concentration gradient is maintained. So in that case you can see a hypotonic solution, there is a hypertonic solution. Okay, and then there is a isotonic solution. So these are the three cases that you will find. Okay, so in all of these cases, so what exactly is hypotonic or a hypertonic or a isotonic is, this one is, you will find water. So let's talk with respect to water here. So for all this we'll talk in terms of water. Okay, just a second. Yeah, so in this case you can find more concentration, like more water you can find. So that is more water and less solute. Okay, so here it is less solute. So in hypertonic, what is the condition? Less water is there. Okay, and there is more solute. So this is with respect to hypertonic condition. Hi Chetna, good evening. Okay, so in that case, So you can find less water and a more solute. All right. Now isotonic solution, when we mention, so same concentration, that is in and out. So let's see what exactly happens, like how exactly endosmosis, exosmosis occurs in this session. Okay, so now let's see this part. So let's see, this is with respect to animal cell. Okay, so an animal cell, what would be the condition? So we have a beaker now. So let's say a plate maybe. So this is a beaker is kept. Okay, so in this beaker, what exactly do we have? So in this beaker, so let's say there is a hypotonic solution. Okay, so there is a hypotonic solution. That means what? More amount of water is there, less solute. Okay, and we have placed the animal cell there. So let's take RBC is kept. Okay, so this RBC is kept here. So now you can see, so in this RBC, it is a hyper condition. Okay, so this is with respect to hyper. So let me just write it hyper here. Okay, so what will happen? You can see the movement of water from their region of a higher concentration to their region of a lower concentration. So in that case, the water which is there, the water will start to move from the hypotonic solution to a hypertonic solution. So what exactly is happening? So there is a endosmosis process. Okay, so why do we call it as endosmosis? Because the movement of water you can see inside, so that is from outside to inside, and that's the reason we call it as a endosmosis. And this cell is basically RBC itself. Okay, so same way now exosmosis, if we have to mention. So let's take a beaker again. 1 second. So now exosmosis we will see. So now this is as usual, this one is a beaker. Then as usual we have kept a RBC there. So this one is RBC. So in RBC, what condition is maintained? In RBC, the condition maintained is hypo. Okay, so here it is a hypo condition, and in the beaker it is a hyper condition. So now the water which is there inside, so this water, you always know that the water will move from their region of higher concentration to their region of lower concentration. So in that case, the water will start to move out. Okay, and because of the water movement, what will happen? So there is a exosmosis process that you can see. So exosmosis is basically the water is moving out, that is across a semi-permeable membrane. So that is their region of higher concentration to their region of a lower concentration. So always remember that whenever the water movement is seen, so it is always from hypotonic solution, okay, to a hypertonic solution. You can see a movement. Is this clear? Any doubt in this? All right. So now with respect to that, so let's see what is plasmolysis and other things. So this is done, osmotic pressure. So now what exactly is osmotic pressure? So this is the minimum pressure that must be exerted. So this is to prevent what? So in order to prevent osmosis, okay. So this is to prevent osmosis occurring. So there is a pressure that is exerted by a component of a cell. Okay, so that can be cell wall. So it is going to prevent the osmosis, and that we call as the osmotic pressure, or you can just say here osmotic pressure of a solution is a measure of its tendency to, that is tendency to take in water by osmosis. Okay, or we can also say that it is nullify the osmosis. So osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution. Okay, when the two are separated by a semi-permeable membrane. So whenever we are talking about the osmosis, so always there is a role of a semi-permeable membrane that can be seen. Okay, so then other than that, so we'll see these questions later. So I'll just explain some of the terms that are mentioned, so you can see this one. Okay, In this one, what is given? So again, this is taken from the Selena textbook itself. Just a second. Yeah. So this is taken from the Selena. Alright. So you can see here, this is kept in an animal. So there now this is for an animal cell. Where it is kept, it is placed in the isotonic solution. So what exactly happening? So water entering the cell equals the water leaving. So there is a water that enters and there is a water that leaves. So that means the balance is maintained. So we can say that no movement, or the term which we can use is, so water entering the cell equals the water leaving. So this we can see in a isotonic solution. So previously I told you when it is placed in a hypotonic solution. So this is with respect to RBC. So now you can see that the water entering the cell is less than the water leaving it. So water is not entering. So lot of water is actually leaving it. So that is mainly because surrounding condition is actually hyper. So we have kept it in a hyper condition and the cell is in a hypo, so you can see the exit. Okay, so that means all the water starts to come out. Now hypotonic solution, when we mention, so there is a water movement, so more of water movement you can see, and that's the reason the cell is going to swell up. So same thing with respect to plant cell. Okay, so plant cell, I need to explain you one thing. With respect to plant cell, if we mention, okay, so let's say this is now a plant cell having its own cell wall, then there is the cell membrane and other things are present. Okay, so now here in this case, what exactly happens is, so now this is a vacuole is present. So let me just draw the vacuole also. So there is a vacuole. So 90% of the plant space, like volume is covered by a vacuole itself. Okay. So now this condition is normal condition of a cell. Now if I keep this particular cell in a hypertonic solution, okay, so I am keeping this in a hypertonic solution, what will happen? So here inside it is hypotonic. Okay, so here the solution here is hypotonic, that is low concentration you can find it. So water will start to move from hypo to hyper. So as you know that the cell wall is already rigid. Okay, so this is rigid. So that means it will remain intact, but the cellular content along with the cell membrane, so it will come to a corner. So now as that happens, so let's see the structure of a cell wall. So let me just draw here. So now this is a plant cell as usual. After it is being kept in the hypotonic, now as the water has started to move out, because hypo to hyper movement you can see. So the from the cytoplasm the water will move out, from the vacuole the water will move out. So in that case you can see that the cellular content along with its vacuole, so this has come to one of the end, that is one of the end of the cell wall. So in that case now this space is completely free. So cell wall has still remained the rigid itself, but the cellular content what is there along with the cell membrane, so it has come to this position because there is a water loss that has occurred because it was kept in a hypertonic solution. So what exactly happened? Exosmosis occurred here. Okay, so as the exosmosis occurs, so you can see all the cellular content is coming to the corner. So in that case this has remained intact. So now this process we can call as a plasmolysis. Okay, so why do we call it as a plasmolysis? Because there is a lysis in the plasma, so that is the cytoplasm has been lysed. So why do we use the term lysis again? Since all the water content has come out, like only a little might be there, but rest like earlier because of the inward movement, that is endosmosis had occurred. So this was in a distended condition. The cell membrane was in a distended condition, that is expanded condition. but as we kept it in a hypertonic solution, so you can see there is a exosmosis that is occurring. So since there is a plasmolysis, the plasma has been lysed because of the water movement outside, and you can see the cellular content is coming at one corner. Okay, so this you can see, so this process is called as a plasmolysis, and the condition of this cell is, so it is a flaccid cell. Okay, so we call this cell as a flaccid cell, or a flaccidity also we can mention. So this is the condition. So this one is a process where the cellular content is coming down, like coming at one corner, and the cell wall has anyway remained intact itself, and this condition of a cell we call it as a flaccidity. Okay, now we'll do one thing. We can keep the cell in the hypotonic solution. Okay, so what we can do, earlier what we had done is we had kept the plant cell in a hypertonic solution, and you could see there was a exosmosis that was occurring. Okay, so now what we will do is we will keep this same cell in a hypotonic solution. So now we are taking this cell and we are taking a beaker again. So let's take a beaker, and in this beaker let's put up a hypotonic solution. So here you can find a hypotonic solution, and as usual what do we have now? So now the cell wall, I mean this cell is actually it is plasmolyzed, that is in a flaccid condition. Now, so same cell we are keeping here. So this is a plasmolyzed cell itself. So cellular content has come down and the vacuole is here. Okay. So now after we keep it in a hypotonic solution, as already here it is a hyper. Okay. So here it is hyper and here it is hypo. So what will happen? Always the water will move from their region of higher concentration to their region of lower concentration. That means the end osmosis can be seen here. Okay, so this one you can see a end osmosis process occurring. So let's have a look at this process now. So once the end osmosis occurs, so you can see again we'll take up a big like the normal cell only. Let's draw the normal cell here, just a second. So in that case, one second, yeah. So now as the endosmosis has occurred, so all this water which is there, like as the water has moved in, so inward movement has occurred. So again you can see a distension in the cell wall. So that means now this cell membrane is expanding, or we can say distending, and this vacuole is also taking up its shape. So that means now this cell is actually in a turgid condition. And what is the process here? So here it was a plasmolysis process, and here deplasmolysis. So I'll just change the color, so that is to highlight it. So now this is a deplasmolysis process, as you can see that the plasmolyzed cell has returned back to the normal condition. So this one is a vacuole, and again it has got its shape back, so that is the turgidity has been achieved. Okay, so in that case there is a plasmolysis, there is a deplasmolysis, so flaccidity and turgidity that you can see. So that is based on what type of solution it is being kept in. So there is a endosmosis and then there is a exosmosis process that can be seen. Okay, so now this cell is turgid, and because of its turgidity, so it has regained its shape. And now we'll see one thing, so that is what exactly is wall pressure and what exactly is Turgor pressure. Okay, so I hope the flaccidity, the turgidity, all these terms are clear. Any doubt in this? Okay, so now let's have a look at what exactly is wall pressure and a Turgor pressure. Okay, so this is basically wall pressure, we'll see. Okay, and then there is a Turgor pressure that we need to see. Okay, so now the cell wall is present. So let's draw the cell wall. So now there is a plant cell. So this one is a cell wall. Okay, then you can see there is a cell membrane that you can see. Okay, So this one is basically a cell membrane which is completely distended. Okay, so here what exactly is happening? So as the endosmosis has occurred, because the moment endosmosis, that means water has moved inside this plasma membrane. So this is what, this is wall, that is cell wall it is. Okay, and this is what, this is a cell membrane. So that is in case of a plant cell. Okay, now let's see what exactly is Turgor pressure and what exactly is the wall pressure. Okay, so now as the endosmosis has occurred, so this cell membrane will distend, like it is expanding. So let's say that it is expanding further because of the movement of water inside. All right, so this is further expanding. Now you can see that the cellular content. Okay, so cellular content is putting a pressure, like it is applying a pressure on the wall. So in that case, this will put up a pressure on the wall. Okay, so this we call as the Turgor pressure. So let me just label this. So this we call as the Turgor pressure. So what exactly is Turgor pressure here? The cellular content is putting up a pressure on the cell wall. So that we call as the Turgor pressure. So now, in order to prevent, so now, as the water keeps on moving inside, so there is a chance that, if the wall is not putting up a pressure, so in that case, the cell will burst open. So as you all know that the cell wall is already rigid, so this wall will also put up a pressure. So that is the, in order to prevent osmosis. So in that case we can say that, so it will try to put up the pressure on the wall, that is cellular content. So wall is exerting a pressure, so that is on the cellular content. So that is to stop osmosis, so that no more water entry is there, and there is a chance that if the wall pressure is not there, so this cell can burst open. So here what is happening, the wall pressure is maintained. So wall, that is cell wall, is exerting a pressure on the cellular content, so that we call as the wall pressure. So in this case, the Turgor pressure and the wall pressure, so these both need to be balanced. Okay, so only if a balanced condition is there, so this cell will remain in a turgid condition. If, okay, so here let's see this, if Turgor pressure, okay, so this is if Turgor pressure exceeds the wall pressure. Okay, so this is with respect to exceeds it. Okay, so if the Turgor pressure exceeds the wall pressure, so in that what we can see, so basically the plant cell will burst. Okay, so there is a chance that the plant cell will burst open, and that is how you can see the fruits will open up. So this will burst open. So this is because the Turgor pressure is exceeding the wall pressure. So you can see that the cell will burst open. So same thing happen with respect to the animal cell also. Only thing is that there is no wall pressure. So as the Turgor pressure increases, so as much as possible the cell membrane will expand. So still the endosmosis is occurring. So in that case, it will burst open, and that's how we can see there is a bursting of a cell that can be seen. Okay, so here if a Turgor pressure is exceeding, so this is exceeds the wall pressure, so the plant cell will burst open, and all the cellular content is coming out. Any doubt? Is this clear? Okay, so here let's see this, like what exactly is active transport. So we need to yet to discuss, we are yet to discuss this part. Okay, like we already said, so if a mineral has to be taken, okay, so in that case energy has to be used, because the cell membrane, what is this? So that is the semi-permeable membrane. Okay, so what exactly happens there? Semi-permeable membrane does not allow the solutes to enter inside, like that is why we call it as a semi-permeable or a partially permeable. So why do we use that term? Because it allows only the water to enter, it does not allow the solute to enter. Okay, Now but a cell, when we mention, it does require, cell does require minerals also. So in that case, uh, hi Siddhi. So is this Siddhi from Unacademy class 9 plus class? Siddhi Gupta, I don't know. No, okay, so then I don't know, I guess. So maybe previous class I have seen Siddhi name. I thought that is the Siddhi. Okay, all right, no problem. Anyway, I remember the name Siddhi who attends the class. All right. So in that case, active transport is basically passage of a substance. The minerals are taken inside now. So how exactly these minerals are taken? So let me just explain you this. 1 second. So now cell membrane is there. All right. So let's say this is a cell membrane. So cell membrane is having a protein. Okay. So this will have a cell membrane protein over here. So let's say this is a protein which might be present. So now if a mineral has to be taken. So let me just draw it in a different way. So I'll just expand this one. So let's say this is a cell membrane on the whole. Okay. So this one is the cell membrane. I'm just drawing a bilayer because that a semi-permeable membrane or a cell membrane when we mention. So this is actually a bilayer itself. Okay. So that is not just one layer will be there. So two layers you can find. So in that case, now let's see. So there is a protein over here. So which is just like this. Okay. So this protein structure is like this. Okay. So in that case, if a solute has to be taken, so in that case, so for active transport, so this is with respect to active transport. So now in this case, so there is a transport that has to occur from low concentration. So you can see this definition. Okay. So active transport is passage of a substance, so that is a solute. Thank you so much, Siddhi. So this really helpful means a lot. Yeah, that yes, all always we always every educator wish for your good itself. So we take all the classes whenever we are doing. So this is basically keeping in mind that so we'll be discussing different questions. So keeping in mind that this is gonna help you for your boards. Okay, so that is mainly with respect to your final exam. So this is every educator wish the same thing. So all the best. Okay, you can perform well in all your exams. Only thing is basically you need to work hard and work smart. All right. Okay, so let's see this. So active transport is basically passage of a substance. So that is from lower to higher concentration. So using the energy, so that is the term. So that is the keyword that you need to use. So in that case, you can see that the solute has to move in now. Okay. So in that case, energy has to be used. Why there is a ATP requirement? Because naturally what will happen, the movement is always from high concentrated area to a low concentrated area. So here in this case, just a second. Yeah, so here in this case what is happening, the movement is occurring from low to high. Okay, so it is low to high. So in that case what would be the condition? There is always a requirement of ATP. So in that case, let's say the solute is gonna attach here. So now this has to be transported inside. So now this protein can turn up. So now this has to be taken inside. So in that case, let's imagine, so not imagine, so this is how it works. So that is by taking in a solute. So how exactly ATP is used up? Now there is a protein. So this protein is carrying this particular solute, a mineral. Okay, so now this is gonna get reversed like this. So this will just move in the different direction, and you can see this particular. So now the same thing is actually moving. So that is by using energy. So that is ATP. So in that case, the solute which was there on the outside, so this is now coming in. Okay, so that is how. So now this whole thing is actually a semi-permeable membrane that I have drawn here. Okay, so inside the semi-permeable membrane the solute is taken, so that is by allow, like proteins, certain proteins will allow the movement. So now you can see that the solute which was here on the outside, so that is low concentrated area, and here it was a high concentrated area. So but by spending energy we are taking in this solute, so now this solute is transported inside. So that is how you can see the movement. So active transport, when we mention, it is the passage of a substance from its region of a lower, from its lower to higher concentration, that is opposite to what happens in diffusion. So that is using energy, so that is from the cell, so through living cell membrane. So active transport is a direction opposite to that of a diffusion. What happens in diffusion? So it is higher to lower, so this is in a diffusion, so this is high to low. Okay, but in case of active transport, what is happening? Low to high movement you can see, and because of this low to high movement, so you need to spend the energy, always you can see the water, so there is too much of water. Okay, so water will flow down from high region to low region, so there is no water moving from low to high because already there is less water, so there is no movement occurring. Okay, so naturally what will happen, always from high region to low region only movement can be seen. But if I want the water to move or a solute to move from low region to high region, so in that case energy is required. Okay, so in that case we'll be using the energy and the transportation can be done. So that is about the active transport, which is mainly done for the solutes. All right, any doubt? Is this all clear? So now cannot pass through the cell membrane of the root cell, so this is because their concentration is higher, so forcibly carries inward, so from the region of lower concentration, so that is what is mentioned to the, this requires ATP supplied, so that is in the form of energy. All right, so let's have a, so we saw all of this, what exactly is Turgor pressure, so what exactly is wall pressure. So this is, osmotic pressure is also maintained, so that is as the as we can say there is a maintenance, basically wall is exerting a pressure, the cellular content is exerting a pressure, the Turgor pressure, so water moves into the cell, so due to the osmotic concentration of cell content, so in that case osmotic pressure is also there, cell wall restrains the expansion of cell with the wall pressure. So in that case, so there are three forces that are acting, the the wall pressure and the osmotic pressure, so that there is no much of a water entry that can occur. Okay, so let's okay, so we are done with today's session, so any doubt you have you can ask. All right, so before I end this session, so let me just say about this. So for class 10 ICSE Premier League, so 31st I have a session, so that is IPL, so that I'll be discussing on absorption, so some absorption by root, so that I'll be discussing in 31st session, so that is 5:15 p.m. on Unacademy Learners app. So you, all you need to do is download the Unacademy Learner app. So we'll be discussing it on 31st 5:15 p.m. So we'll be discussing 3 marks or 5 marks question here. All right. So then gladiators batch we have, so this is a all subject batch. So if you are subscribing for class 10, so you will be getting class 11 and 12 subscription absolutely free here. So there is a UNSAT, which is a offline test for IIT, JEE, NEET, UG, and as well as for class 9 and 10, which is conducted across 40 cities. All right, and it is conducted on June 4th and 5th. So last day to register is June 2nd, so you can use the code Vani YT, okay, to get 10% discount for the enrollment. So for enrollment it is 199, so you can enroll for this one, and you will stand a chance to win basically, you know, the iPhone. So top 40 rankers are going to get the iPhone here. The test takers also get the Unacademy t-shirt, so you can go for this one. Okay, so basically scholarship is the one where you will be getting a scholarship at Unacademy Center and online subscription, so you are basically getting a offer them. Okay, so introducing a community, so never miss on any update from your favorite educator. Get session notes and practice material there. Share your feedback directly with the educator via poll. So you can find this, like, you know, my community link in the description. Okay, so description you can just check in there, so you can find in the link, and the notes of this one maybe I can send it over there. Okay, so Bugs Bounty, so this is opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Okay, all right, so we are at the end of this session. Okay, so see you all in the next session. So you can click on bell icon to get notified about the classes. So subscribe to our channel, that is Unacademy ICSE and ISC boards. Like and share the video and download the Unacademy Learner app. So this PDF I can send it on my community. All right, so see you all in the next session. Bye bye everyone, thank you so much.